In this tutorial we will discuss the PCA and regression module. As you can see, a project has already been loaded, and the HTO demultiplexing, quality control and gene selection modules have been applied. During the gene selection, we performed two analyses. One with the VST method, and one with the mean var plot method. The objective of this module is to carry out a PCA, a principal component analysis. This analysis will allow us to transform the space of the genes that have been selected, to give the space of the principal components, which is more representative of the heterogeneity of the data. To perform an analysis, we must first select a list of genes. We can start from the complete list of genes in the project, which contains, as you can see, 18,244 genes. We can also choose the list of 1,000 genes selected by the VST method, or the list of 447 genes selected by the mean var plot method. We will start with an analysis with the list provided by the VST method. We must first choose the number of principal components to retain in the final result. The value 50 is a commonly used default value. We also need to give a name to this analysis. It is important here to give a representative name because we will have to find the result of this analysis in the successive modules. We will therefore name this analysis PCA 50PC, then the name of the list used, VST 1000. We then run the PCA analysis. When the analysis is completed, we obtain several pieces of information. First of all, we obtain the ShIVA plot, which gives the principal components on the x-axis, and their standard deviation on the y-axis, which represents the quantity of information carried by each principal component. In the second tab, we have access to a heat map. There is a heat map for each of the principal components. This heat map allows us to see which genes are carried by the principal component, which we call loadings. The yellow color shows overexpression, the purple color shows underexpression. So the first genes are those that have a positive principal component. The second ones have a negative principal component. The second type of information we have is the correlation table between the principal components and the covariates. It is indeed important to understand whether the variability detected by the PCA is due to biology, or to elements that could come from a technical or experimental effect. In this table, the columns are the principal components, and the rows are the covariates. We find the percentage of ribosomal genes, the percentage of mitochondrial genes, the number of features, and the number of UMI in HTO and RNA. The color represents the expression value of the Spearman correlation. Blue represents positive correlations, red represents negative correlations. The size of the bubble represents the statistical significance of the correlation. If we observe a strong correlation between a component and a covariate, for example PC4 with the number of RNA features, we can go to the scatter plot, which is on the left, and select in X, PC4, and, in Y, the number of RNA features, to observe if this correlation seems real. Here we observe that, although there is a trend, this does not appear to be a significant element of technical bias. If you find a correlation in your data that can hardly be explained by biology, but rather has a technical or experimental origin, then it is possible to eliminate this bias by performing a statistical regression of this covariate. To do this, simply select the covariate in question in the Select Variable to Regress Out field, or even select several covariates if you observe several inadequate correlations, and then run the PCA again. The analysis will then be performed on the regress data. We can now run a PCA again, choosing not the list of genes provided by VST, but the 447 genes selected by the mean var plot method. We still choose 50 principal components and change the name to reflect the chosen list. We then run the PCA. As you can see, the results obtained are different from those obtained with the VST1000 list. It is indeed common for gene selection methods to give different or even very different results. It is therefore interesting to perform PCA analyses on different gene lists to see the impact on subsequent analyses. Once the PCAs have been performed, we can move on to the dimensionality reduction module, tsne and UMAP. This module is the subject of another tutorial video.